Hello there, welcome back to the channel. Today I want to share with you a really interesting discussion that I had with a scientist. His name is Utkarsh a Ph.D. graduate with quite an impressive education and experience in molecular biology. Because in our previous meeting we talked about many captivating subjects, I invited him over to discuss more about his education, his PhD thesis and experience in US. Stem cell research and advances in biotechnology were some of the subjects we talked about, so check out how it went. Hi Utkarsh, thank you for coming. I would like to start the discussion by you telling us a little bit about your background and where did you study?
Okay. I am from India, from New Delhi, born and brought up. And after I finished my high school in Delhi, I moved to Bangalore to carry out my bachelor's in biotechnology. I pursued BSc in biotechnology and I studied various aspects of biotechnology, microbiology, and some biochemistry. So this I did at Bangalore University. After I finished my bachelor's, I moved further down south to VIT University, where I did my master's. And this was also going more into depth about biotech and microbiology, and also learning various aspects of how biotechnology industries function. And when I finished my master's, what I realized is that even though I had interest in a lot of topics, stem cells definitely caught my eye. But I realized that before I was ready to make a commitment to do a PhD, I wanted to gain some background of or some hands-on experience of what it is to actually work in a lab.
Why did you choose this field and what motivated you to pursue PhD?
I have been always a very inquisitive and a curious person. I find it very natural to keep asking questions and wondering about the what and the how and the when. And research in general seemed like a perfect fit for somebody to satisfy their curiosity. And ever since I was in high school, I started thinking about specifically biology and the kind of complexity it had to offer. I was very interested to understand how systems work, how nature functions, and how different parts of the body function. So this led me in a direction to generally have an understanding towards biological systems. And that's what sort of took me to biotechnology, because that is a field that allowed me to study a bit of both basic biology and some applied aspects of it as well.
Mm-hmm.
So, how it works in India usually is that we have a lot of theoretical knowledge in universities, but the extent of practical knowledge is rather limited. So, before making a decision of whether I would like to pursue a PhD or not, I had started working in a stem cell institute in India, in Bangalore. And during my 4 years of work that I did at Institute of Stem Cell Biology and Regenerative Medicine, I learned a lot about how it is to work in a lab, how it is to conduct projects, how it is to train younger students. So it was a great experience for me to learn all those tools and techniques, and most importantly, learn to ask the right question. So that was something which was very key to my reasoning for selecting to do a PhD. Because at the end of those 4 years, I was so sure that PhD is such a natural step for me to pursue that I just knew that I have to specialize in a certain topic after my master's, and that's how I decided to do PhD.
And PhD, did you pursue it in India or you went somewhere else?
So while I was carrying out a project in Bangalore, I encountered a certain topic of RNA biology. And it was while I was studying RNA biology, I realized that I wanted to be in a more international cosmopolitan environment, because it's essential to be in the midst of a rich scientific community, because scientific discussions trigger good ideas, and those ideas also help you network. And then I started looking for places which have a cohort of RNA biologists, and that's what brought me to Vienna.
What are the three problems that molecular biologists are hoping to solve?
Wow. Okay. So this is certainly not an exhaustive list, but I think everybody has on their mind how to cure cancer. That's definitely been on everybody's mind. The second thing is about certain neurodegenerative diseases such as Alzheimer's, Parkinson's. We know that there is a long history of patients who may or may not, because of any genetic history, might develop certain conditions. So, of course, molecular biologists, immunologists are interested in understanding what's going on there. And at the same time, there are other rare diseases which may not be as prevalent in all ethnic subtypes, all children, or only adults. So there is also an active effort on trying to identify rare diseases earlier on in its stages, so that we could start developing clinical therapies for that.
Okay. And during your university and basically during your studies, what was the most interesting project that you were working on?
Right. When I was still in Bangalore and I was trying to understand, or I was very fascinated by the idea of stem cells, because stem cells were or are a very hot and cool tool that has been researched over the last 2 or 3 decades or so very exhaustively. And that's when we learned that a single cell has the potential, a single stem cell has the potential to develop into any other cell type in the body. I mean, our body has more than 250 cell types, and how a stem cell is able to differentiate or specialize to form a certain type, only a liver cell, for example, or a cardiac cell, or a brain cell, a neuron. So, how all of this is achieved is something that is very interesting for me, and I learned dynamics about how such specifications happen early on in the stages of development using both mouse models as well as human cells. And this was something which was amazingly cool for me to understand. And of course, when we go deeper and deeper into the mechanisms, both cellular and molecular, you tend to uncover more and more unknown, which is very, very relevant to our current understanding of stem cell biology. So that is something very, very interesting for me.
So basically, you can research and research, because we don't have right now, we don't have all the answers, what they do, how they do it.
Yes, we do know as to why a cell behaves in a specialized way, but we do not know how, when, or how does the specialization occur. So there is a concept of epigenetics, which sort of controls the expression of genes in different tissues. Like, for example, every cell in your body has the same genes, but there are mechanisms in play that have been uncovered after decades of research, which have led us to the understanding that there are certain genes which are functioning only in the neuron but not in the heart, only in the liver but not in the bone, and so on and so forth. So we know that yes, every cell has the same genetic makeup, but what is actually controlling the expression or the specialization of a certain cell type to behave in a certain way, we understand that. But we do not necessarily understand why that is the case.
Okay.
Yeah. So, yes, we do know a lot about it, but certainly not enough about it. Having said that, we have made enough progress to be able to use stem cells in therapy. There are patients who have certain spinal disorders or cardiac disorders. So their stem cells have been harvested from their body, reprogrammed or conditioned to function in a certain way, which can sort of help the patient cope with whatever condition that they are currently in. For example, there are patients who suffer from who are quadriplegic or biplegic, where basically either two or all four limbs are not functional. So scientists and researchers have harvested stem cells from their body, processed it in cell culture, re-injected it back into their spine, and a lot of patients have, in combination with physiotherapy, recovered some limb movement. So people who were absolutely, for example, even born paralyzed or became paralyzed as a result of some kind of an accident have been able to regain movement, and that is because of stem cells.
That's amazing. That's really amazing.
So stuff like this. At the same time, there are patients, kids who might have some cardiac issues. So as a result of efforts of many molecular biologists, geneticists, we have been able to sequence their genes, identify possible mutations and the conditions that those kids might be carrying, harvest their stem cells, process them in culture, and inject it back, and you can see those cells to actually work in the heart. You can see some of those cells actually beating in a petri dish. If you look at into a microscope, you'll be able to see cells beating in it. So that was one of those moments for me when I was pursuing stem cell biology, that man, this is the thing, that this is something is so amazing. Yeah, okay, we can read stuff in books, but seeing is believing. So that's how I sort of try to.
So basically, then you realized, I love what I do.
I love what I do, and there is nothing that's gonna stop me from pursuing PhD, because it's for a scientist, it is not just the curiosity that drives what we do. It is also a question of generating knowledge. If I work on something and I'm able to generate a piece of data that can make the scientific community think and see that, or utilize this data or put it in context in some other biological system, it invokes ideas. And that's how science functions. That you need to see that you are not just pursuing science, doing stuff in a test tube, but actually generating knowledge from it and invoking thoughts and ideas in the scientific community. So that's something which was very, very, like, one of those facts that were in my mind when I had also decided to pursue PhD, like my doctoral studies. Yeah.
And regarding PhD, what was your thesis about?
Okay, so our cells have genes. Genes are in DNA. So DNA makes protein. This transition goes via another intermediate molecule which is called as RNA. You might have heard of RNA a lot these days, especially because of COVID-19, that the genome of the COVID virus is actually also RNA. So the idea is that when genes start getting activated, there are certain parts of the gene which sort of splice together. There is a part which is coding and there is a part which is non-coding. There is a process called splicing through which two coding parts come together to be able to form a functional protein. At the same time, in RNA level, there are some mutations that can occur. And these mutations at the RNA level are usually called as RNA editing events. So a single point mutation in those bases has the potential to interact or perturb how the splicing landscape occurs. So the essence of my thesis was to understand how splicing and these mutations at the RNA level go hand in hand. And if at all, an interaction between these two processes can have an impact on gene expression on a tissue specific level. For example, if the interaction can lead to expression of a gene in bone marrow cells and not in the neurons, or vice versa. So this is just for example. And so my thesis was essentially trying to understand the dynamics of these two processes, and for this I had worked both with mouse tissues, mouse cells, and also some human cells. Yeah.
And this was the research was done in the lab, and then you wrote about it, and as I understood as we talked before, you also published it. Can you tell us more about this?
Sure. So, how usually science works is that you ask a question, you conduct experiments, and then you try to either, based on the results of those experiments, you try to tell a story that if things make sense. And at the end of this is a part where we have to communicate our results to the scientific community. And how this communication occurs is by publishing. So we write our findings in the form of a research article, and this is now sent to a scientific journal, which now accepts the paper for peer review. That means that the paper will be sent out to other experts in the field. They would try to go through the data and then try to say whether this actually makes sense, or there is more scope in the project, and or there are still things which needs to be done to make the story more sound. So I was able to successfully do this, and I published two of my papers, two of my main research findings in a scientific journal called Genome Research.
I know that basically somebody has to fund your research, yeah?
Right.
How is this handled? How is this treated in academia versus in a corporation, in a company?
Okay. So in academia, since I come from academia, I can talk more confidently about that. How I understand is that a group leader would write up a scientific project which would be based on certain hypotheses and existing literature in the field. From that, they will try to identify these are the missing pieces of important data missing in the field. So, first part is to identify what's missing, then write up a scientific proposal, and then this scientific proposal is now submitted to various funding agencies. For example, in Austria, the main funding agency for academic research is FWF. And what we do is that the boss would, or established group leaders would write up these scientific proposals, submit it to the funding agency. Now they would also review it and also send it for peer review in the to the experts of the field. See if the project has legitimate grounds for being funded, and based on their assessment and our how much money we have asked, how many people we would need for it to employ for this particular project, how much amount of experimental material we would need, how much amount of expertise we would need to outsource. So based on these kind of factors and the feasibility of the project, the funding agencies determine whether a project should be funded. And because all this research funding is ultimately a part of a country's budget, it's a part of a certain percent of the GDP of a country's budget. So of course it's limited. So it's very, very competitive. So you have to be really at your A game to be able to get there. In industry, on the other hand, they have a different agenda. There, the idea would more be to do applied research. Because in academia, you generally have more fundamental biology addressed and more like at the interface of academia and industry. But at the industry level per se, they are more funding research which will definitely or most likely lead to an application. So there is a different process in place for them to get funding for that. They can be private investors, they can be venture capitalists, and at the same time they can also be funding agencies specifically funding either startups and or industries. For example, I think in Austria there is also the FFG, which is specifically funding industry-related projects.
Cool, cool.
Yeah.
That means, so you're saying that in the end, the product, it can be a product or an application, mostly for the corporation.
Exactly, because in industries, they do not necessarily care about publishing results because they want to make a product, trademark it, patent it, so that they can know that this is their baby that they made and they can capitalize on it. Versus in academia, it's more about publishing your results in scientific journals based on how you publish, which journal you publish. That brings you different kind of laurels to your CV.
Can you actually convince during your PhD that some research is helpful for some certain company and actually ask the company to fund your research?
So, the project that I particularly worked on is still fundamental biology, and it wasn't anywhere close to being used in an applied setting. But it did give us a lot of new information about how we should think about certain molecular processes when we think about gene expression or complexity. So, it kind of added another iota of complexity to the system. It added to it. Yes, that's how essentially it is. But it wasn't a particular application that could be sought out for funding.
Okay. Now, turning back a little bit, you were talking about stem cells. What would be the one thing that normal people, would know about the stem cells? So, what should they know about it?
Okay, so stem cells are cells which have the capability to differentiate into any other cell type. Let's call it like a master cell from which all other cell types can be derived. Now, there is a very common myth in stem cells that a stem cell is derived only from the embryo, and if you kind of try to isolate cells from there, you might be perturbing the human embryo, and this sort of has led to a lot of ethical guidelines that research teams have developed to sort of balance what is progress and what's not. So that takes us a bit into a more philosophical aspect of research. But in stem cell biology, the power of stem cells is such that if we are able to understand how different cells are derived from one master cell, in future we may not even need any transplants to be conducted. Because usually the problem with transplants is that you have to have a genetic match to your closest relative, closest family member. If at all not, then there has to be, the other closest match as possible, and there is always a risk that your body might reject it. There is host versus graft disease, and all that effort, amount of money that you put in for doing those expensive surgeries, and not just the money, but the amount of chemicals and drugs that your body has to go through during that process is not necessarily a good thing for your system. So what stem cells offers is that power that that the scientists are able to use stem cells from your body. That means it is your genes, and I process it outside your body in a dish, do my magic, and throw it back in, or inject it back, surgery, however, and then your body is able to cope with it. So that either you are able to cope with the injury, or you are able to cope with whatever kind of stress that your particular organ has faced. So there is no chance of your body actually rejecting, because it's your genes. So this is something which is an amazing technology and finding that we have had in the last couple of years.
So, Utkarsh, is it possible to transform one cell type into another?
Yes. In 2010, there was the first study conducted in Stanford University, where the researchers converted skin cells from a mouse, and converted that into a fully functioning neuron. So that was the first evidence for a possibility of what is now called as cellular reprogramming, where you can completely convert one cell type to another. So by doing this, we don't necessarily need only your stem cells, but rather any cell type. So this gives us even more power that I can take any cell type and convert it into another. So this way, we are not necessarily tapping the limited number of stem cells that the body has. And also, an interesting fact is that as you age, the number of stem cells in your body go down. So it is difficult to get more healthy cells when you age. So you rather do this or do something like a stem cell banking, so that you can save some cells while you are still young. There are various stem cell banks that can take adult stem cells and bank it. And in case in future you have a requirement, you can go back to that source and use those younger healthy cells.
One question related to this, because I don't really grasp the idea. So basically, as we age, you said that we don't have as many as when we are young.
Yes.
But all of these stem cells, do we produce during our life, or we have a certain number that is slowly decreasing over time?
So yes, our body produces stem cells, because ultimately what you need to know is that every organ has their own pool of stem cells. One common myth in stem cell biology is that stem cells are derived only from embryos. No, there are also adult stem cells which are present in every tissue. So if there are, for example, a liver organ, they will have a liver master cell. From the liver master cell, they will have different liver cell types, because again, liver also will not have one but many cell types. So there is a liver stem cell population, there is a heart stem cell population, etc.
But this one, so let's say the liver stem cells,
Yes.
cannot produce heart cells.
It cannot produce directly, but you will have to again carry out the process of cellular reprogramming. So in theory, it's possible, but the question of efficiency, the right conditions, etc., needs to be established.
So it's not yet.
It's not a done thing just yet.
Mm-hmm.
Yeah.
Can you tell me now what are the biggest problems in the industry?
Sure. So since I just mentioned something about stem cells which are resident in their own tissues, one of the biggest problems is that when we study stem cells, we usually take them out of the source tissue, study them in a petri dish in cell culture, do our magic, and then we inject them back. So, okay, they may be functional, but they may not necessarily function to the extent that we would hope for. And that's because while they are in culture, they are in a two-dimensional setting. But in their tissue, they have everything around them. It's a 3D system. So these days, researchers are actively working on a next frontier of stem cell biology, which is something called as organoids. What that means is that I can use those cells, those stem cells from your body, and generate mini organs. And using that, studies are being conducted to sort of understand what is actually happening to the physiology, to the behavior of the cells in this three-dimensional setting. It gives us a better handle on what's actually going on in the system. So, but at the same time, producing these mini organs is still a process which is underway. We have already established mini brains, mini livers, but other cell types, other organs, and the efficiency still is to be worked on. Another thing which the industries usually face is in getting enough stem cells, because okay, we can take sample from a patient once, twice, maybe thrice, but we cannot repeatedly go back to the patient. So we should be able to somehow multiply these cells in culture, because we need more amount of material to begin with to be able to conduct our experiments, to be able to use that.
But you cannot use animals for this?
We can, but then there are always differences between mice and humans. And it's also a question of expenses. It's also a question of, yes, even though mice and humans have the same genes, exactly the same genes, and a lot of them function exactly same, but a lot of them also do not. So there are similarities, yet there are differences. So while we are doing the investigations, it would be better to do it in the system that we hope to use. That's the idea. And that's why also researchers in industries are trying to optimize certain bioreactors or fermentation technologies so that they can multiply these stem cells in big bioreactors and then hope to use these cells for conducting experiments.
Okay,
Yeah.
it's very interesting. I was reading a book last year about how we can achieve immortality.
Right.
And basically now we are moving forward with biotechnology more and more. And the question is, can we achieve this, or what is the limit that our body can take?
So I cannot answer whether we can achieve immortality, but I can tell you this, that through the work done by immunologists, molecular biologists, geneticists, we are already on the way to delay aging. There is a lot of studies which have been done in various model systems, not just in humans, but using plants, using zebrafish, using mice, using worms, trying to understand at various trans species level that what's actually going on. There are worms which if you cut, they completely regenerate. So there are a lot of mechanisms that have been understood in how these regenerative properties are, and they are also now being employed in studying human aging. And yes, there are some studies which are done, and that's why you also have sort of these anti-aging products on the market. There is a lot of organic anti-aging products, cosmetics, which have been there, which sort of help your skin look younger or wrinkle-free, etc., etc. So there are studies which are done because we understand how certain cells behave when you condition them. So I do not know again if we can achieve immortality, but we can certainly delay aging.
Utkarsh, please name one breakthrough that made humanity go one step further.
Okay, so in the last decade or so, there has been work done specifically from two main researchers, one group in the US and one in Europe, where a technology named CRISPR-Cas9 has been developed. This technology has given researchers the power to change genes at their will. For example, if there is a condition like sickle cell anemia, beta thalassemia, where we know there are certain genes which are not functioning, CRISPR has given us the capability to easily, if I may add, go and remove the piece of faulty gene and replace it with a functioning one, so that we are able to now hope to achieve the function that the gene is intended for. And this is not just possible in one cell type, it has been established in amazing number of species. It has been done in fish, in pigs, in humans, in mice, in rats. So we are now in a position to very easily change your genes. So this has given us so much amount of power. We had other previous technologies to do something similar, but the efficiency of that was extremely poor. But with this technology, now it is so easy. You can see CRISPR-Cas9 almost on every biological paper that gets published out there. It's so easily spotted. And as a result of this amazing finding, the two scientists which were basically responsible for this finding were also awarded the Nobel Prize in 2020.
That's cool. Yeah. You were mentioning in our talks about your experience in US. You were at a conference.
Right.
Can you give us more details about your experience?
Sure. So I had the pleasure of attending an international conference hosted by the RNA Society. So that society sort of has an annual conference, and I had the opportunity to present my PhD work in the conference. And so that was the first time I spoke in front of almost 1,000 people, and it was a great experience to learn about to be on a stage that big in front of experts of every field. It's pretty unnerving to get up there, talk about what you want to achieve or what you have achieved in your work. And so this was a conference that was held in University of California in Berkeley. And yeah, so my work had been shortlisted to for an oral presentation. So you can either choose to either give a talk or present your work in the form of a poster. So my work was shortlisted for a talk, and yeah, I had given a 30 minutes presentation at the conference, and I got a really good response after that.
And what did you talk about? Was this part of your thesis?
Yes. So essentially, I had summarized the major findings from my PhD thesis.
Mm-hmm.
Yeah.
Super. Now let's go further to another topic. So, and this is like hypothetically speaking, if you would have enough money and the power to do any research, what
Yeah.
would you do?
Oh my God. This is an amazing question that I have no answer for, because there is so many cool things out there to pursue, whether it is respect to disease or whether it is respect to basic biology. There is literally tons of amazing, interesting questions. But one thing that I would be really interested is to develop something which is called as an artificial intelligence drug. Because what I think about these days is that the amount of information that we have out there, whether it is about computational biology, artificial intelligence, I would like to somehow combine the power of both of them and sort of use machine learning algorithms to establish or let the machine predict targets that might be relevant for drug development, and or use the same algorithms to identify drugs which may be more effective than the currently known ones. So I would really hope one day to see what I would call as an AI drug, like an artificial intelligence, completely identified de novo by the machine. Now we use this information to work in the lab, try to see if we develop it, and if it can actually do the work as predicted by the machine. So that would be something amazing, and I would happily invest in a startup or in a setting like this to generate something which is called as an AI drug. I know already that companies are trying to use something like this, but it is too soon, because of course we are still in the very infant stages of even knowing enough about artificial intelligence and how it works.
What are you currently reading?
I recently have started reading Barack Obama's Promised Land. So I've just read the preface so far. I also heard him on Audible, and it's just great to learn about how his 8 years in the White House were, and how he thinks about politics, and not just politics per se, but also to inspire people to think about different ways of democracy. So
Okay.
that's something which seems amazing. Yeah.
And what is your favorite book?
I don't have one, but I can recommend one book which will actually also help the readers to get a general understanding of molecular biology, not in terms of the science, but in terms of the thought behind it. There is a book by Siddharth Mukherjee. It's called The Gene and Intimate History. That's the name of the book, The Gene and Intimate History by Siddharth Mukherjee. He's a stem cell biologist, currently I think at Columbia University. His books are amazing. I would highly, highly recommend reading this book.
And would it be understandable for me as a non-scientist?
Absolutely, absolutely. Like I said, it does not have too many scientific details to divert the reader, but it actually tells you in the form of a story how things were developed. How did we start thinking about genes? Who were the first people who actually started observing certain things which led us to where we are today? So that's something told in a very storytelling way of saying things rather than a research article.
Now, at the end of the interview, I want to give you the chance because I bombarded you with a lot of questions. You can ask me any question and hopefully I can answer.
Okay. What is your general opinion about how scientists are?
So,
Do you think they are always geeky, nerdy, or do you have some sort of a stereotype?
Yeah. So I think it depends also on what area. So really, I see scientists, of course, so they are dedicated a lot of their time to research. Yeah, so they are really focused on one thing, and I think that if you really focus on one thing, you can achieve great things. So basically, from this point of view, I really admire them. I also read some books that people were really going into one topic for their whole lives, dedicating their lives, so even putting it as the main priority of their life to this, even if in the end they didn't achieve what they were hoping to achieve. You know, these people are pushing us forward. So I really appreciate, of course, their work. And yeah, I mean, you have to be a special kind of person to fit into this,
Right.
yeah, into this path. Yeah.
Right.
So thank you so much.
Thank you very much
And
for having me. It was
yeah,
a pleasure.
thank you as well. And I wish you all the best in your career, and hopefully you achieve what you are looking for. That's all, folks. Thank you for watching. If you have any comments or suggestions, please leave them below. Until next time, read more and invest smart.